Good morning, biology, and welcome back. This video is going to cover chapters 1.1, 2, 1.3, and 1.4. I'm going to try to get really fancy and link when those start, when 1.2, 1.3, and 1.4 start down in the description. We'll see if I'm successful. But let's jump in. Last week we talked about why living things are alive, what biology is, what it studies, the different kind of topics that we're going to be going through. We're going to continue that line of thinking. In this 1.2, we're going to talk about some bigger concepts of biology. Then we're going to talk about the scientific method, and then we're going to finish up talking about microscopes as tools. So let's start out talking about this picture here about diversity. All organisms are different but share similarities. We can tell difference between species. A monarch butterfly is much different than a scorpion, but they're both inside the Arthropoda family. They're very closely related to each other versus closely related to us or like a tuna or a plant. What this picture doesn't have on it is including like fungus or bacterium, which are all very close to us as not very close, but related to us as well. So to describe the different organisms that we see, we use this phylogenetic tree of life. And we use this to base things based on similar characteristics. So animals, we are things that move, we are things that use energy, we have multicellularity, we're a lot like the fungus that do that, or plants, but we're a lot different than the bacterium over here. So we're farther apart on the tree here. Biology and biologists believe that life comes from a single common ancestor. And before that, we had this primordial ooze or like soup that eventually led to the rise of this organism that eventually differentiated into all the ones that we have now. So as you go down this tree, you're moving closer to that original organism in structure and function, what we think that thing looked like. All right, there's three domains of life. There's bacteria, archaea, and eukarya. And we call these the main branches of the tree. And as bacteria, like these single cell guys here, these archaeans here, and archaeans are really commonly found in really extreme environments, places like the Dead Sea or places like vents around Yellowstone and geysers. And then the one we're really common with is eukarya, things like fungus here and elephants and animals and plants and protists. Underneath the domain, we have what we call kingdoms. And so inside of domain eukarya, we have four kingdoms. There's Animalia, Plantae, Fungi, and Protista. And this here is an amoeba. It's eating this little guy here. It's like wrapping, these are called pseudopods, which means false feet. It's wrapping those little arms up around that morsel there, and then it'll bring it into its body and digest it, which is very cool. There's six kingdoms. The first four are in domain eukarya. There's two others. One kingdom is in domain bacteria, and we simply call it kingdom bacteria because there's only one of it. An example here is this Vibrio cholera species here. This causes the disease cholera, which is a diarrheal disease that's really common in places that have contaminated water. And then one domain is in domain archaea, and this is an archaean here, kingdom archaea. And this guy is commonly found in places like the Dead Sea. He's what we call an extreme halophile, which means that he really likes salt. So we will also be talking, about diversity, we'll be talking about ecology. Ecology is a specific branch of biology that studies the interactions of organisms. So this is like how tadpoles, frogs, flies, fish live together, but also how the quality of water helps them and how much sunlight they get, how much nutrients this is providing for the plants in the area. And when we have these very large communities, we call them ecosystems. And we're going to do some really fun stuff with ecosystems. We're going to talk a lot about ecology when we get to it because it's very, very fun part of biology. You can see a lot of it with your own eyes and like understand it pretty easily. All right, another topic we're going to discuss in this class will be evolution. We'll be discussing how populations change through time. And we'll be talking about this process called descent through modification. And what this means is that genes are passed from parents to offspring, and as they're passed from parents to offspring over and over and over again, just like when you take a single piece of paper and you copy it 1,000 times, it starts to change and look a little bit different than the beginning. And that's exactly what's happening with these organisms. And this explains why our tree of life has branches and how organisms are related. And each of these topics are not standalone. They will each tie into each other and kind of build off of one another. This picture here, we call this group of animals the ungulates, or the hoofed animals. And so these all had a original ancestor who had hooves, and then they have branched out to be really good in their ecosystem. This guy's really good at reaching the tall branches, the giraffe is. Things like our cows are better at like digesting just a lot of grass. The tapir down here, he's common in the Caribbean and Latin American area. Very cute. He's one of my favorites, especially the little babies. They get like these really cute spots. But all of these guys are related, but they've all kind of differentiated and spread out across the globe to be a little bit different. All right, natural selection. This is another big topic. In natural selection, organisms have certain favorable traits that improve their ability to survive and reproduce. So for this example, we're looking at the difference between snowy rabbits and desert rabbits. These are both rabbits. They're very closely related organisms. But the snowy rabbit has a vastly different set of skills than the desert rabbit to survive in his environment. You can see that he's very fluffy. He's very, very short, stout. He's got these little tiny ears. And this is all to help him keep heat in for his body. As a mammal, we produce heat. We're warm-blooded, so we need to constantly keep around a constant body temperature. He has managed to do this by having this extra fur. He also is a very different color than his desert rabbit friend. This is because they're in different environments. He's trying to blend into that snow, so that when a predator, a fox or a hawk or something is flying over or looking over the environment, he's kind of like hiding in there, not really showing up. And then our desert rabbit over here, he's a little bit different. He's stockier. He's got these longer legs and these longer arms. He's probably going to be moving around more. He's probably going to be faster. He's got shorter hair so that he can get rid of that heat energy in this hot environment. And that's also what these really big ears are for. They help him hear, but they also release heat off of there, the same way that you release heat off of your head and feet. So while these two are related, they look a lot different from each other because they have different kind of adaptations to survive in their environment and be good at avoiding predators in their environment. All right, so to jump into 1.3, all the science that we've talked about so far, all of the ecology, evolution, all of that kind of things, we use the scientific method. And the scientific method is just the definition is an organized approach for answering questions. As scientists, we try to answer questions in a way that we can describe them over and over and over again. So it's not just a one-time, somebody saw this and it happened, but it's like every single time we can describe this or get close to or understand more about it. But there's two things that need to happen for the scientific method to work. And the first is the events of the natural world need to have causes. They need to have causes that we can actually understand, quantify, and make sense of. And number two is uniformity. The fundamental laws of nature operate the same way at all places and times. So gravity is going to work the same here in North America as it will in Australia, as it will on the moon. It might be a little bit less, but it's still the same force doing the same thing. And we can quantify this, and it has a natural cause. And for gravity, it's the mass of an object. So the bigger the object, the more gravity it possesses. The sun has much more gravity than the Earth. That's why we rotate around it. So the scientific method is something you naturally do, but we're going to talk about the steps that actually happen inside of it. The first is observation. It's the act of looking at and seeing a natural occurrence and questioning that occurrence. Why is it in wintertime populations of deer come from the high altitudes where it's really snowy down to the lower altitudes? And we can once we've had this question and we've seen this thing, we can form a hypothesis. And a hypothesis is a proposed explanation. Usually you do a little bit of research before it. You see if it's got any scientific backing already. But you're looking at this particular aspect of the natural world and trying to figure out why it functions. So for our deer example, maybe there's not enough food up in the higher elevations. So that's why these deer are coming down to these lower elevations. All right, number 3 is make a prediction. A statement that forecasts what would happen if a test to the situation was true. Well, let's make a prediction. Let's say that I go up to these high elevations and I throw out a whole bunch of hay bales. So now there's a ton of food in that high altitude. My guess would be, my prediction would be, okay, these deer are going to stay up here now because there's a lot of food up here. And then the next step is to run an experiment. This is to test your hypothesis under these known conditions. Maybe you set up game cameras on the top of the mountain there and you record each deer that comes through, versus down low, you have something down there and you also are recording it. If we had lots and lots of deer showing up to the hay bales and having a good snack, that would, we could say probably that has something to do with it. That's why they're coming down from that higher elevation, because if they have that food, they stay up there. So when after you run the experiment, you can analyze the data. Usually we'll be trying to get some numbers from data. We'll be trying to compare it against other groups. And the goal with analyzing data is to say, hey, was our hypothesis supported or was it not? Was our prediction correct? And then the final step of the scientific process, after everything else goes well, is to report findings. Good scientists share information. I know with our corporate setting and a lot of world issues going on, a lot of people are trying to monetize science. But the goal of science and the scientific method is to continue to progress the information and the knowledge that the human race possesses. So we're always trying to get better. All right, so for experiments, it's important when you have an experiment that it's called a controlled experiment. You'll hear me say the only difference between science and messing around is writing it down, but it has some kind of backing. When you have a controlled experiment, you have an experimental group and a control group, and the control group provides the normal standard. So if we're using our deer example still, let's say on one mountain, I put a whole bunch of hay bales on there and we're testing whether or not that keeps the deer higher. And then on the second mountain, I don't put any. This would be the normal group. This would be the group that's not getting any of the experiment. They have the same amount of food that they normally have up there, and we want to see what's the difference between the two groups. Does this change of introducing a food source have an effect on their behavior? And that's a lot of what ecology is. Does this change have an effect on animal behavior? All right, the experimental group should be identical to the control except for one factor. So in this example we're talking about, this factor is the presence of hay bales, the presence of food. That is the one factor that we are changing as researchers and as scientists. We call this factor the independent variable. And the important thing about the independent variable is it can be manipulated by the researcher. So in our example, it's the presence or lack of hay bales. In my example here on this page, it is flowers and light. So plants have what they call phototropism. Phototropism is the tendency towards plant to direct themselves towards sunlight. They use the sunlight as food, so being faced towards that sunlight will give them the ability to grow big and strong. This is why when you have indoor plants, you want to rotate them, you want to spin them around so they continue to grow kind of more in an upward direction instead of just straight over towards the light, because they will try to go straight towards that food source. In nature, this is well, they're doing this because they want to acquire more sunlight, they want to compete for it. But we can change in the lab how much light these plants are getting. We could take this light and we could move it over here, and now we could see if this plant would grow the other way. This is an independent variable that we can manipulate. And then from there, we can talk about the dependent variable. We call the dependent variable also the responding variable, if that's a little easier for you to think of, but it's the thing that's affected by the independent variable. So for the last one, we could do amount of sunlight versus how wide the flower opens, or we could do, in this case, this is amount of daylight hours was done on this plant. This one has many more daylight hours than this one, and they behaved completely different. So the amount of daylight hours affects how these plants reacted, and that's the dependent variable. This one looks a different color. It has more blooms. This one doesn't have the blooms. So just the difference between the amount of sunlight that these guys got affected their behavior, affects how they actually physically look and are growing. All right, the collection of data. Most experiments, you're measuring a variable. Usually it's the dependent variable. You should know your independent variable because you set it. You would set that timer for that light to an hour, to 2 hours, to 4 hours, to 8 hours. And then we could measure how wide this flower is opening up. Did it open up an inch or 2 inches or 3 inches or 4 inches? What we're looking for is we're looking for this thing called quantitative data. And quantitative data is any data that can be measured, specifically having a number. So quantitative data is numbers. Qualitative data, and if you see qualitative data, it just means that something happened. This is like when you do elephant toothpaste. If you don't record anything, if you just dump all the ingredients in, it's qualitative data. But if you instead attach a balloon to it, try to weigh how much gas has been produced, that's quantitative data. And then with the analyzing data thing, the goal is to determine if the data is accurate, if the data is good, if all the numbers are close together, are they far apart, is there any kind of trend that's happening here? And it's just to see, the analyzing data is a big part of, did this experiment produce the results that I thought it was? And for this drawing conclusion part, we're looking at, is the hypothesis supported? Meaning, we can't prove a hypothesis. We can just say, with the current knowledge that we have, this hypothesis still works. It's much easier to disprove a hypothesis, to say, okay, this didn't work, this wasn't effective, nope, we have to try something different. So if your experiment works, your hypothesis is supported. We, as humans, don't create science, we discover it. Science is out there, it's working all the time. Plants are growing, gravity's working way before we understood how those processes work. And they will continue to do so. But hopefully, our understanding of those processes will also continue to grow. All right, making inferences. So a conclusion made on the basis of fact and previous knowledge, but not direct observations, is an inference. So an inference is more like what we usually say when we say, I've got a hypothesis. It's more likely to be an inference. Inferences are not directly testable. So an inference might be say, oh, okay, well, if I go through a black hole in my spaceship, let's see if I can draw a spaceship here. Close enough here. If I go through this black hole with my spaceship here, little boosters there, what's going to be on the other side? Well, I think, based on what we know about science, I think that if you were to make it through the event horizon of a black hole, you would end up somewhere else. This is an inference. Right now, we don't have the ability to go test this. We can't see if this would happen or not. So right now, it's an inference. Once we get more technology and we get the ability to travel and do things like that, we could then test this like a hypothesis. But until that happens, it's called an inference. It's subtle, but it's an important difference. All right, so let's talk about applying results. In biology and science, in this class, we will be making models. These are structures that are created to help represent or describe things that we see in the natural world. So the example here is Mr. Watson and Mr. Francis Crick here. They made a model of DNA with cardboard balls and wire bars to take this really, really tiny structure. There's millions of molecules of DNA inside of your cells. To take these tiny structures and blow them up to be something that they can actually see and manipulate and try to understand better, because it works a little bit better for the human eyes. We didn't have like electron microscopes at this time, so we were using radio and kind of like radio imaging, like X-rays, to figure out the structure of these molecules. So really, really cool. Really, really helped them to have the ability to take these and use these models to look at it a little bit closer. Makes the things manipulatable, and you can actually hold it in your hands and look at it and understand it. All right, so now that we've done the experiment, we've tested a couple hypotheses, we can start to construct theory. And this is when a set of related hypotheses are tested successfully over many times, and there's a large deal of data that can be explained by this. And we classify it now as a theory. And theories are highly tested. They've been shown to, in just about every situation, they're supported. So examples, things like the Big Bang theory. We see that all these galaxies are moving away from a central point. That's really good evidence. We find tectonic plate theory. This is the one where we're talking Earth's plates that we talked about in Earth science are moving around and carrying the continents with them. Atomic theory, where all matter is made up of atoms. We're talking about theory of matter and energy, and cell theory, and the theory of evolution. And we'll talk about cell theory and theory of evolution in this class specifically, and how we found that we haven't found any living organism that aren't cells yet. So as far as we know, to be alive, you have to have cells. All right, communicating. Results of scientific experiments should be shared so that other science can learn and ask new questions. And this is usually done in the form of publishing a paper. And in this way, papers are reviewed through a process we call peer review. And this is where experts in the field are taking these papers, they're reading them, and they're critiquing them. They're saying, hey, you did this part of science really, really good. Your methods here were really, really good. But I'm interested to see, are these results replicatable? So they determine if the experiment can be replicated, if it can be redone, if this happens every time, or if this was just a one set instance. And for it to be good science, this experiment needs to be replicatable. Preferably, somebody could pick up your experiment and run it in their own lab, and it would be the exact same results each time. All right, here's a kind of the last thing we're going to talk about in 1.3. This is a flow chart of the scientific method, and I want you to note we talked about all these steps: this asking question, constructing a hypothesis, testing an experiment. But I wanted to point out it's not a straight line from ask a question to communicate results. Sometimes you get down here and you're testing an experiment and it doesn't work. For whatever reason, your experiment is not getting the information that you're looking for to test your hypothesis. So if your procedure is not working, you'll have to troubleshoot it, check all your steps, and then test again. If that still continues to not work, you might have to change something different. You have to change your hypothesis or change your whole experiment. But even if your thing does work and you analyze your data and you draw conclusions and your data doesn't quite align all the way with a hypothesis, now you've done background research for your new project where you're asking a new question and you run a new experiment and you're getting this new data. And then after you go through that, now maybe the results are better aligned with the hypothesis. Now you can communicate it. So if you were a researcher and you were trying to find a new drug, a new cancer drug, and it does it in 1 out of 4 people, it's probably not well tested enough to use on the public. So you're going to go back, retry it again, and try to get that 4 out of 4, try to get 3 out of 4 better results before you actually publish and start to use this drug. All right, this is the start of chapter 1.4. In 1.4, we're talking about microscopes as tools. A big part of biology is using microscopes to understand the smaller components of that organization. We're looking at cells, cells that build tissues, tissues that build organs. And the one that we're going to use the most is called a compound light microscope. And these are ones that allow scientists to see enlarged images of objects. And how they work is they're going to shine a light through your specimen and then magnify the image so that you can see it. Now, the kind of limiting factor for a compound light microscope is the objects has to be thin enough for light to pass through. If you have really thick, cumbersome objects and then the light can't get all the way through, you won't be using this compound light microscope on them. You'll either have to take a really small slice of that object or use something different. But first, let's talk about the anatomy of the microscope. There are four main parts of the microscope. There is the eyepiece here, which magnifies the image. There is the objective lens here. This is where light is passing from the bottom up in through the specimen, up through this. This further magnifies it, and you can change on, you can see on this one, it has 3 different modes of magnification here. You can change your magnification on this one, which makes the image bigger before you see it in this ocular lens. The stage is this kind of spot here, this black area where you're putting the specimen, and then the light source is the light bulb, or on really old microscopes, it was a mirror that you had to like shine at the light and you had to get it just right, that you use to create this light for viewing the image. So the light goes up through here, into the specimen, up through the objective lens, into the ocular lens where you can finally see it. All right, so now we're going to talk about magnification and resolution, and they are related, but they are not the same. Magnification is the increase of an object's apparent size. So the ocular lens starts at 10 times magnification. And then the objective lens multiplies it further. So if your ocular lens is at 10 times and your objective lens is at 10 times, how this actually ends up working is you multiply these together and you get 100 times the normal magnification. So if you have, let's say you use the 40 times objective lens, instead of getting 100 times magnification, now you're getting 400 times the magnification of just the regular lens. So this is why microscopes are so powerful. They can take things and they can make them 400 times bigger than what you see with the normal eye, which is awesome. Resolution is a little bit different than magnification, because even if you can blow something up really, really big, you need to be able to see it. And resolution is the power to show detail in an image. Think of it, if you're a computer guy or a computer gal, think of it as having more pixels per screen. You have more ability for clarity and you can see things better. And so resolution with light microscopes, usually we can see with really, really good ones, most images can go up to 200,000 times. And we'll use what's called immersion oil, and gives you a little bit better ability. It acts as a little bit extra lens to help you magnify and have that good resolution. There are some other types of microscopes that we won't be using in this class. Electron microscopes are awesome, awesome, awesome tools. Instead of using visible light, which is really, really big, it uses a beam of electrons, which is smaller than visible light, to produce an enlarged image of a specimen. So it's more powerful in both its magnification and its resolution. The pictures are always in black and white, though, because we're not using the visible light spectrum. And then the computer program, we can use them to add color. But this here is a fly, kind of eyeball and face blown up. You can see the hairs coming off of here. You can see the little different lenses going on here. You get very cool images over there. And we can use these to look at really, really, really small things like viruses and bacterium and all of these really, really tiny particles. So we can even look at individual molecules using electron microscopes. There are 2 types of electron microscopes that are commonly used. There are the scanning electron microscope, which is the SEM, and this passes a beam of electrons over the surface of an object, and it can magnify objects up to 100,000 times normal vision. And the second one is the transmission electron microscope. And this passes a beam of electrons through the specimen instead of over it. So how this works is these are going through there. On the other side, there's a magnetic lens that enlarges and projects the image onto a photographic screen. The scanning electron microscope is going to be the more commonly used one. The transmission one is really good in circumstances where we're looking at like tissue. We're looking at cancers, or we're looking at tumors, or we're looking at different disease processes, and we want to see what's happening on those individual cells. We'll use the transmission electron microscope. Both are great tools. We won't be using them. If you really want to get Mr. Greer a birthday present, he would love one, but they are pretty expensive. All right, so units of measurement in biology and in all the sciences, we're going to use the metric system. More specifically, we're going to use the International System of Units, the SI. This is a decimal system and based on powers of 10. So you shouldn't be using fractions in science. Mr. Greer will prefer decimals. And there are base units or kind of categories of what we're measuring and what we expect to find that measurement in. And they are coming up. There are 7 base units. So these are the 7 base units: length, mass, time, temperature, amount, current, and intensity. For biology, we'll really be looking at length, mass, time, and temperature. If you do any if we're doing that plant experiment where we're doing the different light, maybe you're looking at candela. If you're looking at current, you'd be doing ampere. If you're like your whole study is like on electric eels. But the big 4 that we'll be using are length, mass, time, and temperature, which length is in meters, which goes by M. Mass is in grams, which is in G. Time is in seconds. And temperature is in Kelvin. And Kelvin is very closely related to Celsius, and we'll discuss that at a later date. This is the conclusion to Chapter 1.4 and the entire chapter of Chapter 1. So I hope that you have gained a better understanding of what this class is going to talk about, briefly learned about the different topics we're going to cover, talked about the scientific process, and then have a better understanding of how to use microscopes. And we're going to do a lab upcoming where we look at some pond water and the organisms that are common in it, and get you guys kind of some experience on the microscopes. So I hope you learned something, and I'll see you in the next video. Thank you.